Okay, so welcome to this next video on the Human Genome Project, in which we are discussing how to build a genome library. So the motivation for what we're trying to do, remember, is we are trying to sequence these 23 chromosomes, chromosome 1, chromosome 2, all the way up to chromosome 22, and then finally chromosome X. Okay, right, so there's a problem. We can't just sequence these massive great chromosomes. Some of them will be over 100 million base pairs long. So we can't just sequence them directly. So instead what we're going to do is chop them into fragments and then sequence each fragment, basically. Okay, firstly what we've done, we're going to chop them up basically with restriction endonucleases. So I was explaining what a restriction endonuclease is, basically. So they're called restriction endonucleases because they were originally found in bacteria and they restrict viral DNA entry into the cell, basically. Okay, so the sort of archetypal example of a restriction endonuclease is the EcoR1 restriction endonuclease. So I will talk about this for a moment. So EcoR1 is the archetypal example of a restriction endonuclease. And basically it was found in Escherichia coli, as I talked about here. Eco stands for E. coli, so E. coli, and then R1 stands for the first restriction enzyme, restriction endonuclease. Okay, and I want to talk about this because it illustrates the principle of the recognition site. So basically, EcoR1 cuts DNA at a certain recognition site, which is GAA-TTC. Okay, and then the complementary sequence to that is going to be CTT-AAG. Okay, so this is the recognition sequence for EcoR1. So basically, it will go over to DNA, and if it sees this sequence, it will bind to it, and it will cut the DNA. And basically, the way it cuts is it doesn't cut just directly down the middle. Instead, the way it cuts is staggered. So it will cut the DNA like this. It will cut between the G and the A of both strands, basically, because you've got this GA over here. So it cuts here, and hence what will happen is you'll get these so-called sticky ends, these overhangs of the nucleic acids. So this is the sticky ends. Now, there's something very interesting about the recognition sites of restriction endonucleases, which is that they are what is known as base pair palindromic. Now, what does that mean? Well, basically, palindromic. Basically, if we just discuss what a palindrome is firstly, a palindrome is something like Hannah, okay, where if you read it backwards, H-A-N-N-A-H, it reads the same as it reads forwards. So that's what a palindrome is. Now, GAA-TTC is clearly not a palindrome. However, it is what is known as a base pair palindrome, because if you read its complementary sequence backwards, then it reads the same as it read forwards. So if you read GAA-TTC, here's the complementary sequence, and if you read that backwards, GAA-TTC. Okay, so that's what is meant by base pair palindromic, that the complementary sequence read backwards reads the same as the original sequence read forwards. Okay, and you might ask, well, why is that? It's interesting, but why? Well, the reason is that if the EcoR1 enzyme approaches the DNA from this side, it will bind to this sequence. It will see exactly the same, basically, as if it approaches from this side. If you think about it from this enzyme's point of view, if it comes over here, what it's seeing, basically, is exactly the same as if it could come from this angle. So if you rotate this enzyme around 180 degrees, what it's seeing from this side is exactly the same as what it sees from this side. So it means that it can bind to the DNA whether it approaches from this side or it approaches from this side, basically. It doesn't need to be at a certain orientation in order to bind. So that's kind of cute. Okay, right. So we are performing this restriction digest of our DNA. So that's a fancy bit of terminology, basically, for chopping up all of your chromosomes into pieces with restriction endonucleases. The fancy name for that is it's a restriction digest. Okay, now this is where it becomes important to have more than one copy of each chromosome. So in actual fact, what we're going to do basically is, we'll go through how to build a genome library, and then I'll talk to you about the fact that we're going to need multiple genome libraries, basically. Okay, well actually no, we need to talk about this now. Okay, so we're going to need more than one copy of each of these chromosomes, but that's not difficult to do. We can do that by the polymerase chain reaction. So we're going to amplify these pieces of DNA, basically. We're going to need multiple chromosomes. Okay, so we end up with these multiple chromosomes. So let's say we've got multiple chromosome 1s, and they all need to be identical to each other, basically. And then we've got multiple chromosome 2s, multiple chromosome X's. So we've got multiple copies of all of these chromosomes, basically. Okay, and what we're now going to do, basically,